Good afternoon. My name is Julián Grandvallet C, and I would like to welcome you to the first international cycle of creators in science and arts, organized by the Colegio de Investigación Interdisciplinaria en Ciencias Sociales, Red Iberoamericana de Investigación en Imaginarios y Representaciones, Grupo de Trabajo Corporalidades y Tecnologías de la UNAM, Instituto de Formación e Investigaciones Jurídicas de Michoacán, Clínica de Derechos Humanos y Derecho Ambiental de la Universidad del Estado de Amazonas en Brasil. We sincerely thank to the organizing committee and everyone joining us today. It is a pleasure to welcome Professor Marlet Themis Martínez Archundia. She is a researcher at the Escuela Superior de Medicina of the Instituto Politécnico Nacional. Her work lies at the intersection of chemistry, biology, computation, and medicine, with a particular focus on structural bioinformatics and computational drug design. Professor, thank you very much for being with us today.
Thank you so much for inviting me. Thank you.
Thank you. To begin, we invite you to briefly introduce yourself, please.
Yes, sure. So I was graduated from biochemical engineering at Universidad Autónoma Metropolitana here in Mexico. Then I went to study my master and PhD in Barcelona, Spain, at the Polytechnic University. And yeah, further, well, about 5 years later, I went to Paris and I studied there a postdoc. And finally, I came back to Mexico. I was repatriated scientist at the Superior School of Medicine, which is the institution I'm working currently. So,
thank you, Professor. Wonderful.
Thank you.
So, in recent years, we have witnessed a major expansion in computational power and its application across many areas of science. So, from your perspective, we would like to know what is structural bioinformatics and why is it important for understanding and treating human diseases?
Sure. Well, I have always been intrigued about biological systems in general, from simple microorganisms to very complex, like human beings and so on. And what help us to understand this structural bioinformatics is to study mainly structural behavior of proteins, right? So sometimes we design or we perform experiments, but we don't really get information at molecular level, right? And we need to understand much more, for example, interactions that happen in these chemical recognitions, because it's what we really happen in the proteins that we have in our body. So structural bioinformatics help us to understand all these structural insights at this level. So it's really wonderful and helpful, of course.
Perfect. Your academic trajectory combines different disciplines and training contexts, both nationally and internationally. So we would like to know what sparked your passion for this field of study and how do you come to work in this area of research specifically?
Yes, well, actually, I have many questions in my mind, and maybe the most important, or the one that was very interested, for example, how medication can work in our body. We take one medication, and then what happens? How all the processes start, for example, when we take it, and then this chemical recognition start. One ligand, one compound can recognize one protein or some others, and then all these recognitions with yield to a signaling pathway and a chemical reaction in the end. So maybe this was the main question I had in mind. And then I tried to combine chemistry, biology, which is my scientific background, but now with computational techniques, which is great, right? So basically in the lab, we use, for example, docking analysis, and we use molecular dynamic simulations to get more information about how this recognition can happen in our body.
Perfect. So I think that you will agree that when we study things at the molecular level, it's very interesting and it's very complex within itself, but we also deal with the fact that these systems are inside in other complex systems like the cells and those cells inside
Mhm,
tissues and so
mhm,
on and so forth, right? So how do you think that at the molecular level we could
mhm.
also integrate all of these features? Do you think it will be possible to one time study multiple interactions, like millions of interactions, not just like one protein and one ligand, but multiple proteins at the same time?
That's a very important question. Yes, in general, I think softwares and computational programs are really, or they are being improved in many technical parts, but still we're working on. So far, we can, for example, perform simulation not only with one protein, but we can have, for example, dimers, trimers, or some other proteins interacting at the same time. And I want to make a parenthesis that, for example, in dynamic simulations, what we do is we have like a box, right? Our protein is in a physiological solution, right? So it's water, ions. And then, for example, we would like to see the interaction of this protein and one compound, but maybe also other protein will be interacting. So we are talking that many components are playing in this computational box. So far, we are working to have, as you're saying, really maybe not only one protein, but maybe we can have, I don't know, 20 or 30 proteins interacting at the same time. The problem is that could be, or that would cost a lot computationally talking. So it's a limit, and we're really dealing now because you can have you may need like a not only a very good computer, but also you need extraordinary um, uh, computational efforts and so on, so you can really run a simulation that can give you all these details. So we're still working on that.
Perfect. Yes, I understand. And hopefully, with this rise of machine learning or artificial intelligence, do you think these are like tools that will be for sure used in your field?
Yes, is what we're hoping right now. As I'm saying, we can run simulations, for example, in the lab of the range of maybe 200, 500 nanoseconds. And we can get really very good information about the proteins and how they interact. But I'm talking about, for example, proteins that can have 500 residues. So let's think about it maybe like a GPCR, a membrane protein, which is very good. But imagine, for example, a huge protein like the spike protein, right? In SARS-CoV virus. It's just at the trimer in which it's maybe 4,000 residues. So it's like really, really a monster. We can say it, and it's so hard. We can really run a simulation with these conditions, and that's why maybe we can use some other approach, which is called, for example, coarse grain simulations, in which you can really resume, or you can really have only certain atoms of the protein, not all the atoms of the protein, because the computational task is just really very, very, very huge.
Yes, sometimes we have to like cut in pieces our research so we can deal with all of them, right?
Exactly. Yes.
Yeah, I understand. But so the next question is, throughout your career, you have been
Hmm.
involved in multiple research projects and also
Yes.
interdisciplinary collaboration. So currently, could you tell us more about your day-to-day, like today's research and what does your work involve and how do you carry it out?
Sure. Maybe one of the projects or novel projects I've been working these last years is about drug repositioning. So it's about, let's say, we look for new treatments of existing drugs. Okay, so after the pandemic, it was a very interesting topic because let's think that we know there is or there was a health emergency in many countries, right? People were dying or were really in a bad condition. So what we do is, okay, for the existing drugs in the market, we will try to find new uses. And it became very important. We can remember, for example, drugs like ivermectin and some other that really were helpful in some stages of the pandemic. And so maybe, yeah, after the pandemic, we started to work more in drug repositioning techniques. And we have one project, for example, Ciencia Frontera, and it's about drug repositioning techniques by targeting the tetraspanin CD151 with these proposing drugs. And we found, for example, loperamide and glipizide were potential drugs that can really treat, for example, some stages of metastatic breast cancer and glioblastoma, which is a type of brain cancer as well. Yeah, so I guess this has been very interesting to work with drug repositioning techniques.
That's wonderful. Now that
Yes.
you mentioned that you have to simulate this
Mm-hmm.
environment, and
Mm-hmm.
you are able to run around 200 to 500 nanoseconds of
Yeah.
simulation. So for those who do not like understand what this
Mm-hmm.
mean, can you
Mm-hmm.
explain us more a little bit? What do you mean by 200
Sure.
nanoseconds?
Yes, for sure. So let's think, well, always tell my students, for example, let's imagine that we're going to develop a film of your protein, right? So it is very important to say because what we run when we really develop a molecular dynamic simulation, in the technical part, we are iterating one equation, which is a formula Newton's formula, right? So it's going to iterate in order to have new positions so you can have the dynamic behavior of your protein. In other words, you are on the trajectory, you are going to get many files, but you will be able to visualize this file, and it's like a film of your protein. And as I mentioned, you will have this, you will be able to visualize, for example, your protein within one box of water, ions. It can be the protein itself, or it can be a protein with one ligand or two proteins interacting. You will need to answer your scientific question before you can decide which is your needs in these simulations. But it's basically like that, and time is very important part. So I would say that maybe to run these 200 nanoseconds, you would need 1 month to run this simulation. It's important to say yes, like about 1 month, so you can visualize your trajectory. And maybe the computers in very important institutes can run now milliseconds, in a range of milliseconds, because they have a very, very powerful computers. But I mean, like, you need very specific computers with a lot of power.
I understand. Yes, thank you for the explanation. So
Sure.
when you simulate these conditions,
Mhm.
how different do you have to set the conditions if you try, for example, two different proteins? Are the conditions exactly the same? I suppose that you can set the pH or the number of water. So how do you choose what to put there?
That's very important question as well. Yes, let's think like in the same, when you perform experiments, you may have like a protocol, right? It's not the same when you, for example, perform a simulation with one membrane protein, because you would need a lipid
Mm.
bilayer. You will decide, for example, what kind of lipid bilayer. It can be, well, for example, POPC, phosphatidylcholine is very common lipid. So you develop this lipid bilayer, then you will introduce the protein. So we know that, for example, these GPCRs are embedded in the protein, so you need to specify the program, which type of lipid bilayer. And as you mentioned, you need to set pH conditions and, of course, temperature, but most of the time, of course, you use physiological conditions, no? So it's basically is what we use to run the simulation. But as I mentioned, depending your question, your research question, you will decide how can you perform, because also, for example, it can be a question of, oh, okay, I want to know how my protein can be affected, for example, by heat, right? How if
Yes.
it's the temperature is rising, I want to know all this process. Well, you can study that as well. You can set different temperatures, and then you can study this all the changes that happen. Or the same PhD is not that common. I think temperature is much more common. But you can study all these conditions, and that is fascinating because we know that sometimes in the wet lab, it's very hard to get all this part. So simulations and so are very helpful.
Yes, I think that uh, they are two different areas and they are both needed. But one advantage that I will say that
Mhm,
the dry lab has is that
mhm.
you can answer more questions with less time, with less resources. So it has its perks,
Yes,
but it's a really nice field. So
yes.
you talk about a lot of concepts that can relate to biology and also
Mhm,
computer science.
yes.
So now a personal question, how do you perceive yourself? Do you perceive
Mhm.
yourself as a biology that does computation or a computational person that does biology?
That's a very good question. I would say it's the second. As I mentioned, my background is, of course, in biochemical engineering and
Mhm.
so on. Uh, I have always been passionate about science and how to answer questions in this biological field. And a very important part that I want to remark also is that theoretical work or biophysics doesn't have to compete with experimental work. We have to work together, right?
Yes.
Sometimes people are just very close-minded and say, okay, no, I do experimental work. I cannot get any information about theoretical work, simulations. But the truth is, in the end, we can work together and we can really solve very interesting problems. So if we look at the journals now, we can see that very good scientific papers, and they have also the simulation part. They have docking analysis, they have all these simulations, and they have the experimental work, and it's a very good way to answer questions, right? So yeah, I think that's
Yes.
very important to remark.
I agree. I agree. So, as a biologist or
Mm-hmm.
as a biochemist that studies also computation,
Yes.
what do you think about the people that I'm sure you have came across along your trajectory, either with coworkers or
Mm-hmm.
students? What do you think
Mm-hmm.
about the people that come from a computer science background that
Mm-hmm.
want to do biochemistry? How do they do?
Oh, well, that's also very interesting because I think in science, as we know, it's every day is a challenge and we have to learn many things every day. So it's possible. I would say maybe, um, well, in general, some people start with a theoretical work and so on, and they can perform some experiments. Uh, but you have to have this knowledge. I tell my students, for example, doing this computational work is not click. It's not only click, and it's very important remark. We have to understand all the theoretical background is there. Doing simulation is not only, using a software and set the conditions. We have to understand all the part that is behind. So it can do, as you said, you can start whether, being a scientist, like doing wet lab experiments, and then you can okay, I can start doing these simulations, which I know many people that have done that, and it's possible to do part of computational work.
Of course. So I do think that as a scientific research being constantly an evolving process, what are the next directions or questions you would like to explore in your research?
Well, I would like to continue with this drop depositioning techniques, of course. I think they are now quite interesting. We would like also to continue with this scientific project of Ciencia Frontera I mentioned. We are on the first stage. And right now what we're doing is once that we identify these compounds from the repositional studies, we will prove them in the experimental part. So we will perform experiments, in vitro experiments. We will try to test them on cell lines, breast cancer cell lines, glioblastoma cell lines, and we'll try to see really if they have an effect, an inhibitory effect on these cell lines that will give us really much more information. So we need the experimental part as well.
Yes, of course, but I think that repurposing will be very useful because those compounds they are like already approved and have been
Exactly.
shown to not have any uh, adverse effects or and they can like you can even go buy them today to any drugstore. So
Exactly.
that sounds really exciting. But
Yes.
how do you deal with this problem that sometimes people from outside the computational world that see simulations,
Mm-hmm.
how do you see this concept that these people, for example, say that no, they are cherry picking,
Mm-hmm.
that because you can model anything, but
Mm-hmm.
maybe
Mm-hmm.
like you can have different results if you modify the
Mm-hmm.
temperature or you are like as you mentioned at lipid bilayer. So how can you keep this scientific reproducibility of the model that
Mm-hmm. Oh, that's very important question as well. In general, as I mentioned, we also follow scientific protocols, like when we do experiments, right? So there are some set conditions depending the nature of the protein. So it's not the same if you are, for example, simulating one enzyme, one catalytic enzyme, let's say kinases or some others, or when we are simulating, for example, a membrane protein that I told you about, right? When we need a lipid bilayer and you have to embed it, the technical condition is different. The treatment is going to be different. Also, the simulation time is different because, for example, let's think that soluble proteins, that these kinases or these enzymes tends to be much more flexible because they are just in solution. Let's think they're just in the cytoplasm of the cell. So the condition is, or the flexibility, let's think about that, is totally different from the membrane protein, which is embedded, right, in the membrane, in the cellular membrane. So the behavior is different. The protocol will be different as well. Uh, Not only the conditions, the time also is depending. The more complex proteins you are talking about, the longer simulation you may need it. So that's important remark. How you validate, it's very important. In general, what we do is we compare our structural results with other simulations. That's very helpful. When we get a simulation, we can get statistical parameters. We can say structural parameters as well, such as root mean square deviation or root mean square fluctuation. So we get these values, which are mathematical values that tell us how is the behavior of the proteins. When we get these numbers, we can compare with the previous publications, right? So we say, okay, the values look similar. We can really validate the work from that. And also, of course, we can also do some replicas. We can run, instead of only one simulation, we run at least 3 or 4. right? And then we see the behavior. If it's so similar, then we are validating the work, and we'll see that actually it's how the proteins are moving in the environment, or how is the behavior, no, in one trajectory. So it's more or less how we validate our work.
That's great. Okay.
Yes.
Okay, I get it. So, I think that a huge problem in the field of structural biology for years has been problem of protein folding theory, right?
Oh, yes.
So
Yes.
when this AlphaFold algorithm came out, I think that
Yes.
everybody freaked out and said, "Okay, we got
Yes.
them. We solved it." But now, at the same time, as you mentioned, the experimental people kept working
Mhm.
on it, and then they found out that, well, protein folding is not a thing that happens by itself. Sometimes you
Mhm.
need other proteins that help this protein fold, or
Mhm.
are multiple components so that it can fold properly.
Mhm.
So, do you think that we will ever be able to also model that, like transition from AlphaFold that says, okay, this is a line of amino acids and then they fold like this, but in
Mm.
reality, these proteins are interacting with this other to fold properly?
Well, I really hope so, and I'm very positive in that. I really hope so. As we know, all these softwares that uh, work in really this protein modeling, for example, AlphaFold, as you mentioned, well, are really now extraordinary. They are really, really very good predictions, and give us information about the folding. So I hope we can be even more accurate. The more and more we're really getting closer to what is the nature of the proteins. And the very good also is how the scientific field has been recognized by this, because we know the Nobel Prize in chemistry was really all about these predictions and protein modeling and all these fields. So I think we're getting closer to really solve this mystery. Let's say. I really hope we can get the best results in the near future. It's my position.
I hope so. Yes, I hope the same. And the one of the most exciting things about this is that yes, it has been used to like try to solve these problems, but at the same time, these new tools have given us new problems and new things that we didn't know they even existed. So that's why we need now more structural bioinformaticians. So for that, my question is, what skills and also conceptual foundations do you consider crucial for the next generation of structural bioinformaticians?
Yes, I would say the first question is to have the passion. I tell my students, if they are not passionate about this, about science, about answer questions, is not, curiosity is the first thing that we have to have as a scientist. Always have, something in mind and trying to reach questions on how to solve problems. So I would say curiosity is the first one. The second, maybe to have this the courage, the motivation to really develop techniques, to carry out experiments of theoretical work, and also to have this perseverance every day, because we know sometimes science can be so frustrating. Uh, it really happens many times that we don't get the results that we want, and we just like, oh my god, so much time to have some results, and in the end we don't get it. Science is like that. It's part of every day. So uh, I guess it's just to, yeah, keep motivated, keep doing all the things, no matter how hard or how difficult it is. And I would say those three things can be maybe the most important.
Okay, yes, I totally agree. You mentioned that sometimes there are like other institutes that could have like some more little bit more of computational power, or maybe
Yes.
people that are specialized, for example, in modeling only lipid
Hmm.
bilayers, right?
Hmm.
So if you want to answer a question about some lipid bilayer, but you have not like a lot of experience, sometimes you
Mhm.
start to make relations with those laboratories, right? So
Mhm.
how collaboration has worked for you through these years of research, and how important do you think it is?
Oh, that's a great question. Yes, science is made of collaborations. It has to be like that. Uh, as you said, sometimes or in many cases, uh, all the projects can be so specific that you may know everything. It's impossible, right? We cannot be specialized just in all the subjects, in all the matters. It's just impossible. Another way to really succeed in the projects or in science is always ask questions to the really expertise people that can really be focused on that, and then can not only you can save time, but you can really get the information you need directly from the expertise, and then you are going to build much more information, um, and that could make you even not only a stronger researcher, I could say that, but also you can have a general picture of what you want from the expertise, right? And it's so important. I tell my students always to collaborate. It's always remember that you don't have to know everything. We have to learn from others. That's very important part of scientists or students. I tell also my students failure is a learning also, right? And it's also a very important note to remind. Uh, they have to collaborate, and you don't have to be shy in science. That is another important thing. You have to ask questions, you have to ask for help, and in that way you can really start your projects and get along with others. And not only it's an amazing part of scientists, because it's not only about work. Sometimes you can get even a personal overview and a wonderful personal experience when you interact with other people. In this case, for example, we have the collaboration of Bordeaux ENP in France, and it's so great to collaborate because our students can go there, they can perform the experiments, they come back and they tell me it's just a wonderful professional experience to learn from others. No?
Yes, totally, because nobody, as you said, nobody's expert on everything. So you just can share no knowledge, and even if you work in the exact same field or try to answer the exact same questions, there are different persons that come from different backgrounds,
Yes,
different cultures. So they might have like a
exactly.
different perspective of the things that you're studying, and they can tell you something that you didn't even realize that was happening, and vice versa. You too can tell them something. So yes, I
Exactly.
also think that collaboration is pretty amazing and very useful here
Yes, and
in
essential,
science.
right? When we're a scientist, we need to collaborate. It's just really essential.
Yes, because non-science is made by itself, because even if you publish a paper by yourself, because I don't know, you have a lot of money and you have your own supercomputer at home, well, yes, but you also have to rely on work that has been made for years before that, so that you can run these simulations.
Exactly, exactly. An important part is to have this feedback, right? Is what we
Yes.
get, because otherwise, if you do all the work, okay, it's not that it's bad, but you need this feedback from others, from other experts, from other. We can learn. I also learn from my students, and that's what is wonderful of science, because you have to be open and we say, okay, it's not a competition. It's more a collaboration. And say, you can learn this, I can teach you this, and you can teach
Yes.
me the other part, no? And that's what is science made of.
Yes, totally. Even between PIs or students,
Yes,
I mean, you might have just some more years of doing this, but we are all here because we like to learn and do science,
exactly,
So, but we are
exactly.
under the cell, like looking for the same purpose. So
Yes, exactly.
I totally agree. So you have been around some different places doing science. And, one thing that people in Mexico have this perception is that everywhere else in the world, science is done better, you know? But sometimes it's just, well, maybe they just have more resources than we do, but I don't think that they have like advanced brains or different
Mm-hmm. Not at all.
way of thinking than us. So, what has been
Yes.
your perspective of being away and then coming back to Mexico?
Yeah, that's a wonderful question. Actually, I would think now it takes years, and we know if we are scientists, it takes years, decades to have your dream job. And actually, I thought I have it now. And it's not only myself. When you build your team, which I have it now, I have my PhD students, have my master degree students, bachelor students. It's like a dream come true because doesn't matter where you are, as you say, you can build every day this part, this scientific team. You may be somewhere else, and if you don't have this connection, because also the interaction with people is so important, then you cannot succeed, right? You can start one project, but then you see, okay, some people are not really interested or they cannot want to continue, then the project is just stopped. And that's also very important that you can feel comfortable in your lab with your team. That's the major keys that you can really give you everything to succeed. So it's not a question of one person. I consider it's depending how you are, how you feel. And in the end, it's just that you're saying, okay, the resources are important. Sometimes we struggle to have them in Mexico, which is true. We cannot deny. But on the other side, you have the team, you have the people there, and that's also very valuable.
Yes, I think so, and I also will agree on that. Scientists are some of the uh, professions who the ones that are scientists can really say that they do love their job and they enjoy coming to the lab or whatever it is, but do their work, even though it can be frustrating, or some things are not like going your way sometimes.
Yeah.
But I think that we do really enjoy do what we do and the purpose that we have.
Exactly. Yes.
So, to conclude our conversation, Professor, what message or reflection would you like to leave with those who are joining us today?
Sure. Well, first, I would like to thank the committee for inviting me. I think it's just a great opportunity to learn. We need more of this, really, not only in Mexico, in some other countries. People can really understand some of the basic scientific concepts. They can really get this motivation in science. Some people are just it's not interested and they don't continue. But it's not that they don't like it. Maybe they just haven't heard about the science or scientific topics. So it's just a great idea and a great opportunity to invite them to this to understand all of these topics. I found it amazing, and yeah, just keep motivated in science. That would be my advice. And there's always an opportunity to learn something different. Always, there's never too late, and we have to keep that in mind.
Yes, learn anything and from
Yes,
anybody. You can learn from
exactly,
anybody, right?
exactly. Yes,
Well, Dr. Marlet Themis Martínez Archundia, thank you very much for sharing your time
thank
and
you,
your experience and your work with us. We invite the audience to follow her on social media and to learn more about her research and ongoing
thank you,
projects. We would also like to thank once again the Colegio de Investigación Interdisciplinaria en Ciencias Sociales, Red Iberoamericana de Investigación en Imaginarios y Representaciones, Grupo de Trabajo, Corporalidades y Tecnologías de la UNAM, Instituto de Formación e Investigaciones Jurídicas de Michoacán, Clínica de Derechos Humanos y Derecho Ambiental de la Universidad del Estado de Amazonas de Brazil, as well as the organizing committee and everyone who joined us today. We invite you to watch and listen to the rest of the interviews in this outreach cycle. Thank you very much and see you next time.
thank you very much. See you.